events associated with each. List the postnatal periods and state the age range for each. Now, the major development events associated with each goes beyond what we're going to actually cover as far as development. So we're only going to cover the embryonic period. It's going to go as far as implanting and gastrulation, and that's all we cover. So you need to know the major development events associated up to that, but there are many listed later in the book that we're not going to cover. So that one is limited as far as the events.
Is it on campus? The learning objectives, I'm sorry.
The learning objectives are on campus. There's the chapter learning objectives. Now, on those learning objectives, it doesn't say what's omitted. What's omitted is only on the PowerPoint slide that I'll get to in a second. Okay? List the postnatal period, state the age range for each, and we'll go through that, and you need to know that. You need to know also the dates of the ranges for those prenatal periods. Describe the process of fertilization. We're going to cover that in detail. Describe the blastocyst, the process of implantation and placental formation. We will cover that and talk about it today. List the 3 germ layers, describe their formation, which is an event called gastrulation. We'll look at that. And list the adult derivatives of each. That's in the table, and I'll make mention of the table. Discuss the formation of the neural tube and the neural crest cells, and I will cover that. List and give the functions of the major hormones involved in pregnancy. This is part out of 28, part out of this chapter, part out of even 18. Okay? We'll be talking about that. So, what's omitted? Most of the chapter, as you would guess. In section 29.1, you go from the beginning until you reach the section called somite formation at the top of page 1088. So you're going to go from the beginning of the chapter to where you see somite formation. The rest of section 29.1 is past somite formation, is formation of the gut and body cavities, limb bud development, development of the face, development of the organ system, and growth of the fetus. We're not going to cover any of those. So we're going to cover the beginning up to and stop. We don't cover somite formation. That's the top of 1088 in the upper left-hand corner of the new edition.
It's not going to be tested.
It's not going to be tested, I'm not going to cover it. You may read it if you're interested, but I'm not going to cover it. Okay? So we're going to cover 29.1 up until you read the somite formation at the top of the page on 1088. 1088. See it right there? Yeah. Cover right up to there. And then omit sections 29.22 to 29.7, which is the rest of the chapter. So we are covering just up to there. And then we're covering that in detail, and then after that, nothing. So that's about 9 or 10 pages.
I'm sorry, what were you covering?
Up to where it says somite formation on 1088. And we'll cover that in class, discuss it, and that's all you're responsible for. It'll have a much smaller number of questions on exam 5 and the final, as you would guess, since we're only covering part of the chapter. Okay? Remember, this PowerPoint is on Canvas, and you can go back and revisit it. All the notes. I got a copy up here.
Thank you.
They were already passed around at the back. Okay, here we go. Let's start by looking at the stages or the parts of prenatal development. These are described in the book. They're very self-explanatory. Prenatal development is divided into 3 stages. It begins at fertilization. We call it a zygote. After that is called a morula or a blastocyst. That's all that it's called. Okay? And it goes from fertilization to 14 days after formation. Okay? We call it a zygote, a morula, or a blastocyst. Okay? The embryonic period, during this time we call the individual an embryo, 14 to 56 days after fertilization. And then the fetal period is 56 days until delivery, and we call it a fetus at that time. Okay? Those are the parts or the sections of prenatal development and the times and names associated with it. The prenatal period is extremely important because if you have any problem during that prenatal period, the, up to 56 days after fertilization, any problem is usually extremely serious. Any problem during this time, if for any reason, is extremely serious. It's the most sensitive time as far as the development of that individual. There's many things that can cause abnormalities. If they happen during those 56 days, big problems, big changes, okay? So let's take the postnatal period. This is after delivery. We have a neonatal period for the first month, okay, from birth to the first month. Infancy from 1 month to 1 to 2 years. They're not too specific about that. I guess you would say it's around the time they get running, walking and running, about that time. Childhood, 1 to 2 years, whenever infancy ended, until puberty. Then that's person that becomes an adolescent from puberty till 20 years old. And then an adult is considered 20 years old until death. The period of adulthood is also subdivided into young adult, middle age, and older adult. It's mentioned in the book. So these are the 4 parts of the postnatal period, and then you've got the adult, which is the longest stage. Okay, let's begin with fertilization. Remember that the sperm are going to be deposited in the female reproductive tract, and they have to swim through the uterus and through the uterine tubes to make it eventually to where the ovulated secondary oocyte is located. Okay? Now remember, when ovulation occurs, what is released is an oocyte that is in anaphase or metaphase 2, and it is hung up at that stage, and it will not complete meiosis until it's fertilized. Just before ovulation, it completed meiosis 1 and kicked out a polar body. You see how small a polar body is compared to the oocyte. Remember, the polar body, I regard them just as a convenient receptacle to get rid of some chromosomes, because you're having just one of the 2 products be a reproductive cell. Okay? Around the time it became a primary oocyte, there were glycoproteins secreted by the oocyte that form the zona pellucida. It's a clear area around it, and it is a barrier that the sperm must get through, and we'll talk about how it does that. And outside of it's called the corona radiata. Those are some cumulus cells that were ovulated with it. They completely surround it. So when the sperm reach it, there's just a few sperm that make it there, and they have to, first of all, break through the corona radiata. That is fairly easy, just using physical force to separate those cells. There's not a lot of tight junctions, they're just loosely associated. Okay? When they reach the zona pellucida, the anterior portion of the sperm encounter a receptor, a molecular receptor in the zona pellucida. When that receptor, it binds to the tip of the sperm, it releases enzymes that are stored in the acrosome. And they don't show any loose sperm, just the first part of the sperm is called the acrosome, and there are some stored enzymes at that location. Those enzymes are released only when it reaches the zona pellucida. They're not needed to get through the corona radiata. Okay? Everybody understood where we are now? We're past this division, past this stage. It just shows you when sperm are right there. Okay. As soon as the sperm break through the zona pellucida using those enzymes, it's fairly easy to do, then it encounters the plasma membrane of the oocyte itself. So now we have the plasma membrane of the sperm and the plasma membrane of the oocyte. They hit a receptor, and then the head is brought in. At that is the moment of fertilization, when the first sperm enters the egg. Polyspermy needs to be prevented. You don't want more than one sperm to enter the egg. If you do, say for example, and one more sperm comes in, both of those sperm will form a pronucleus, a nucleus, and the egg will form. They will all unite, and you have a trisomy individual. In the human genome and most other advanced animals, you must have a certain number of copies of each gene and unique chromosomal balance. So having that extra genome, not just one chromosome, all 23, results in an individual which will not survive very long during development. The uterus is set up that if it's abnormal, you're going to have a miscarriage. So the uterus and the embryo is going to be rejected. Rarely, only rarely in the billions of births on this globe has a triploid individual ever made it to term. So they're going to be a miscarriage sooner or later if you have polyspermy. So this egg, once it's fertilized, must prevent polyspermy. There are two methods. The first thing it does is to open some ion channels and to immediately depolarize it to prevent any other sperm from attaching and being able to fuse. That's called the fast block to prevent polyspermy. Okay? The oocyte is then going to release water and some molecules that inactivate that receptor that the sperm contact to release the acrosome to reach the egg. So we're first going to make it so the sperm can't fuse with the membrane, and second, we're going to prevent any other sperm that haven't made it that far from ever being able to get through the zona pellucida. Okay? A fast block and a slow block to prevent polyspermy. It's very important.
The slow block is through water and molecules?
They release water, and it's the
Mostly.
molecules. They release some water and molecules that inactivate the receptor in the zona pellucida that I talked about on the previous slide. Sperm break through the corona radiata and bind a receptor of the zona pellucida. We're preventing that. We're going to destroy that receptor, so if a subsequent sperm reaches it later in time, they can't bind that receptor and release their acrosomes to get through the zona pellucida. They're making the zona pellucida an impermeable barrier to further sperm. Okay? The first block just prevents them from uniting with the membrane. The second block makes the zona pellucida an impermeable barrier because they can't release their acrosomal enzymes, because the receptor to do that is not there in the zona pellucida anymore. So that is what happens. Okay? We now have fertilization, and that would be right here. The one sperm is now just made contact. Okay? The sperm head is the only thing that goes in. It's going to form a male pronucleus. Remember that the oocyte was hung up at metaphase 1 of metaphase 2 of meiosis. So the secondary oocyte is going to complete meiosis 2, form a second polar body, and form a female pronucleus. So we have a male pronucleus and a female pronucleus. They unite, and now we have the zygote, a fertilized egg, officially. Yes, it was fertilized when the first sperm head went in, but the actual forming of the genetically unique individual, in this case a diploid, because we didn't have polyspermy, that would be the zygote. A fertilized egg at this, where the pronuclei have fused, is called a zygote. Yes?
Do the polar bodies do anything?
The polar bodies do nothing. They don't have enough cytoplasm to do anything. They're just barely any cytoplasm with the nucleus. As a result, some people say they divide, but they have so little cytoplasm. You can see their size. That's the whole cell. That's the whole cell. Okay?
Is the zygote only single cell?
The zygote is single-celled.
So once it divides, it's not a zygote.
It's not a zygote. The zygote is only the single-celled individual. And this, since it's a unique sperm and a unique egg, it's a genetically unique individual. And with the number of genes and the number of mixing of the genes for forming the gametes, this is almost undoubtedly a unique individual. Unless you have an identical twin, there's nobody else that has the genes that you have. So this is a genetically unique individual. Just like each of us are in the room. Anyone here have identical twins? How many of you do have an identical twin? Oh. And we'll talk about how identical twins occurs on the next slide. You probably already know. So, yes. So how can we tell you and your identical twin apart?
Just our names.
You mean your twin could walk in here and we still call you in? Okay. What's your twin's name?
Angela.
Annual? Angela. Got it. Okay, unique. Identical twins are uncommon. How many of you are just twins? Okay. Fraternal twins are much more common. Okay, here we go. Now, we start with a zygote, and the zygote is a one-celled individual. That cell, one cell, is going to divide. The divisions occur approximately every 24 hours or so. The divisions, now normally a cell divides, and each one of those doubles in size and then divides. That's not what happens here. We have a cell that divides into two cells, and they don't increase in size. They divide to 4 cells. So we have progressively smaller cells. They do increase slightly in size, but you can't double. There's not enough nutrients available to double the size before you do it. The ovum, what's the secondary oocyte, is large enough you can see with your naked eye. And these are getting progressively smaller. So, but now, each of these cells, for the first few divisions, somewhere between 16 or 8 and 16 cells, all of those cells are totipotent, which means they can form every tissue in the adult. So if these two become separated and each forms an embryo and an individual, you end up with identical twins because they're formed from the exact same cell. Are there differences between twins? As we learn about genetics, yes, there are some differences between even identical twins because there are some events that happen to the genome that can happen differently in different individuals. Plus, there are things that are just developed kind of randomly, like fingerprints. So you and your identical twin have different fingerprints and footprints too. Okay, so the first division, the first few successive divisions are called cleavage because the cells don't double in size before they divide, they just start dividing. And so the cells get progressively smaller, cleavage, and eventually they're going to form, I back up. So everybody understand, even at an 8 or even sometimes at a 16 cell stage, those cells are totipotent and can form, if you separate it, they can form a complete individual with every tissue, okay? After somewhere around between the 8 and 16 cell stage, you end up with cells beginning to differentiate, and they can no longer form every tissue in the human. They can still form lots of tissues, just not all, and they would be called then pluripotent. Okay? Now, do you understand? Was there a question?
No, after 16 cells is pluripotent.
Yeah, roughly.
Oh.
It varies for individual cells, okay? Once you reach 16 or more cells, it's called a morula. A morula is a ball of cells, solid ball of cells, and that's on the next slide. After about 5 days, and by 5 days it is beginning to get close to the uterus. It's got fertilized over here, and it's been making its way slowly through the uterine tubes, heading toward the uterus. about day 5, there's a reorganization of the cells, a rearrangement of the cells in that solid ball of cells, and they're going to form a hollow sphere, fluid-filled hollow sphere. This is called a blastocyst. That's what begins implantation, and that's what we have basically inside the uterus. And the cells forming the outside of the ball are called the trophoblast. There it is, trophoblast, right here, okay? So that's the trophoblast. And green, that's the trophoblast. Inside, in one location, is a small mass of cells, making it thicker at that location, and that's called the inner cell mass. The trophoblast is mostly going to form other structures besides the embryo, mostly. And the cells of the inner cell mass form most of the embryo. Okay? And we'll talk about some of those extra embryonic membranes are. So, we're going to begin with this blastocyst, and it's going to be involved in implantation. And we'll talk about implantation next. I'm pretty sure that's next. Do you have a question?
I thought that total potency was only up until about 6 to 8 cells.
It varies for cells in there, but it's somewhere in that 8, plus or minus a few divisions. There's no hard, fast rule because different cells lose their ability to be totipotent at different times. So it's a range. Okay. Pardon? Okay, let's go and look at implantation. Implantation of the blastocyst and development of the placenta is the next topic. So here we go with implantation. There is a movie that's going to start running as soon as I click the slide. So this is the wall of the uterus, this is the blastocyst, and it's going to implant. Or maybe I have to click the slide. There it goes. And it's going to push in and eventually be all covered by the wall of the lining. When implantation occurs, it's going to embed and burrow itself into the uterus and to be completely contained. And there's a layer of cells over the top. It's the uterine cavity. And if I go forward and back again and go forward, it'll run again when I touch it. See how it burrows in? There it is. Okay? So implantation occurs because the trophoblast right here, this part of the trophoblast, is going to divide and produce some more cells. And those cells it produces do not separate the nuclei. They don't form cell membranes. So each of those is an individual cell. This is essentially one giant cell with many nuclei. So it divides and gets bigger and bigger and bigger. And it's this part of the trophoblast is called the cytotrophoblast, not on the slide, but on the diagram. And this part is called the syncytiotrophoblast. It's a syncytium where you have a mass of cytoplasm with multiple nuclei. The cells divide, but just grow bigger and don't separate the nuclei into individual cells. This syncytiotrophoblast, in order to burrow down in there, has to secrete enzymes that separate cells and break down cell membranes. And you don't want those enzymes to act on the syncytiotrophoblast. So they secrete enzymes that break down the cells, separate cells, break down blood vessels as it burrows in. That's what it does. Okay, as it's breaking down blood vessels, we're going to get blood forming little pools in the area. And the syncytiotrophoblast is going to push down, and the cytotrophoblast is going to follow it down, and you end up with these chorionic villi between pools of blood. These are just ruptured blood vessels. And so we end up with pools of blood. They're going to be fed by an arteriole and drained by a venule. And these are called chorionic villi. The next step is they're going to have cells that go and form a blood vessel going down. And eventually you have, oh, I forgot to mention this last time. After this embedding occurs, the walls of the uterus, the two walls, fuse, so you have no more uterine cavity. The walls of the uterus fuse completely, so you end up with this embedded embryo surrounded by solid uterus. There's no uterine cavity after implantation because the walls, the two walls just fuse together. Okay? So, a little later, a covered syncytial trophoblast. Oh, the trophoblast at this region, at this time, it's the cytotrophoblast that does it. It secretes HCG, which acts like LH and maintains the corpus luteum. Okay? To maintain the endometrial line, maintain the entire uterine line at this point. Yes.
What's the pathway the sperm go to meet the egg?
They have to go from essentially at the cervix up to the distal part of the uterus, then out the uterine tubules.
Oh, wow.
That's the only way to get there.
That's far.
And then the egg goes back on that one, the developing fertilized egg goes back on the same path. Exactly the same.
The HCG, isn't that what they use in the pregnancy test?
They detect this in a pregnancy test. That's what they detect. And it's produced by these cells right there. Cells of the cytotrophoblast. Okay, and we're going to have these lacunae of blood and the chorionic villi go between it. Chorionic blood vessels are going to form in the trophoblast and go down in here, and eventually you have this. This is what a placenta looks like. Now, when the embryo embeds, you're going to form those chorionic villi, like we saw here, around the entire embryo, 360 degrees, and around this way up here. The entire embryo is going to be surrounded by these chorionic villi. So this is much, much later, obviously. But here we're going to realize only one region develops the placenta. All the rest of the chorionic villi degenerate. One location forms the placenta. The placenta looks like this. Here is some arterioles feeding a lacuna and some venules draining that lacuna. And in it are capillaries coming off of the veins and arteries in the umbilical cord. So these capillaries are immersed in a pool of maternal blood, fed by arterioles, drained by venules. And there's a very thin membrane between them. So this is the lacunae and the maternal blood. This is the capillary containing fetal blood. And there's just a thin layer of the syncytiotrophoblast in the basement membrane, which is called the chorion, over which nutrients and oxygen have to diffuse this way, and waste and carbon dioxide have to diffuse that way. Thin membrane. Okay? And this, right there, develops from this. Get a blood vessel in here, we got a syncytiotrophoblast, and we're gonna have a blood vessel there. It's a very thin layer. But the placenta, all the chorionic villi develop all around it, just one location is gonna form the placenta. And the chorion is one of those extra embryonic membranes, and the chorion develops from the syncytiotrophoblast and the cytotrophoblast, which are not part of the embryonic, the inner cell mass, they're part of the trophoblast. So that's an extra embryonic, outside the embryo, membrane. Formation of the 3 germ layers is called gastrulation. Now, just before this point, the embryo was 2 cell layers thick, okay? And it was a disc, called an embryonic disc, okay? And it was just 2 cell layers thick. This was the epiblast, that's the hypoblast. In the hypoblast was 1 cell layer. It divides and gets a second layer, then a cavity forms between those 2 layers. That cavity is called the amniotic cavity. That's the amniotic cavity that the embryo or the fetus is going to be in. Right now, that is going to, well, right now, only the blue right there is gonna form the embryo. Right now, only that blue layer is gonna form the embryo. So this amnion is, which is this layer right here, is an extra embryonic membrane formed from the inner cell mass. Okay? The other cell layer is called the hypoblast. The hypoblast, some of these cells are gonna migrate around the blastocele cavity and form the yolk sac. The yolk sac is extra embryonic also, not part of the embryo. Only the epiblast right there is gonna form the embryo. But we now have an amniotic cavity. How does the embryo get completely in the amniotic cavity? Want to know the secret? What's gonna happen later is gonna just draw these two edges together. Then it's all inside. It's just gonna take and draw them together. Now we have it. The yolk sac in many animals contains nutrients. This contains very weak nutrients. Okay? I think I've covered everything there. Oh, the yolk sac is a source of blood vessels, blood cells, and germ cells. Germ cells that are gonna form the eggs and the sperm. So here it is a little bit later in time. So here's the epiblast, there's the hypoblast. At gastrulation, we're gonna take one cell layer, the epiblast, and make 3 cell layers. Okay? This is the epiblast, that's the hypoblast. The hypoblast cells are gonna be displaced and no longer important. What happens is, around day 13 or 14, gastrulation occurs. The embryonic disc, which is this layer of blue epiblast cells, kind of elongates, so it's kind of long. Then there's going to be a depression that's formed up by one end, and that's going to be the primitive streak. And that, where it forms, it's closer to one end than the other. The end it's closer to is gonna be the cephalic end, the head end of the embryo, and the caudal end is gonna be the tail of the embryo. So there's a primitive streak formed, and it's closer to one end, which becomes the cephalic, the other one's the caudal. And in that primitive streak, we're gonna take the single layer of cells, they're gonna make more layers, then cells are gonna go down through that primitive streak and form 2 layers beneath it. Okay? They all start at the epiblast, they move down, they go down really far, they become the endoderm. They go down halfway, they become part of the mesoderm. They stay up in the epiblast, it becomes the ectoderm. Those are the 3 primary germ layers. We form from one layer, the epiblast, we form all of these 3 layers. Okay? The hypoblast that was there, those cells get displaced and they're no longer important, they don't form parts of the embryo. Okay? Just those 3 layers. Those are 3 germ layers because those layers, by those cells in those layers, based on their position, form certain structures in the embryo. Even though there's just a few hundred cells there, we know exactly what tissues, cells in that layer, and that layer are gonna form. And that's right here in this table. Please make sure you look at this table. This is all ectoderm, mesoderm, and endoderm. The ectoderm basically forms the short list, epidermis, and your nervous system. Okay? There are some glands too and things like that. So, the ectoderm, the skin, and the epidermis of the skin, not the connective tissue, and nervous system. The endoderm is going to form the epithelial linings in your digestive tract and your respiratory tract. That's basically the short list. You can read more detail. And the mesoderm forms lots of other structures. Dermis, cardiovascular system, all the cardiovascular system, muscles, bone, lots of other tissue and organs. Okay? So gastrulation is very important because if you don't form the proper layers, you can't form a lot of these structures and tissues. When this is, gastrulation is occurring, forming the three layers from one, a connecting stalk attaches it to the, this is separating from the uterine wall, and a connecting stalk connects all of this to the uterine wall. Okay? That connecting stalk is going to become the umbilical cord. Blood vessels are going to form through here. Another extra embryonic membrane is going to form for that. Okay? Another structure that develops next to the primitive streak is the notochord. The notochord is a structure that identifies a vertebrate. Every vertebrate has, as far as the classification, biological classification, every vertebrate has a notochord. Not every vertebrate has vertebrae, but all of them have a notochord. A notochord is an important structure because it forms and it induces the formation, causes or induces the formation of other structures. The notochord is a trivial part of the human. Parts of the centers of a few vertebrae is all it is. But the notochord, when it forms, it's kind of rod-like right there. The notochord then causes other structures to form. That process is called induction. Invertebrates do not have a notochord. Not every vertebrate, by the way, has vertebrae. So what's up next? The nervous system. Remember, the nervous system develops from the ectoderm. It's a flat sheet of cells. Yes, it's thick, getting thicker, but it's a flat sheet of cells. That flat sheet of cells is called the neural plate. Here's the notochord that's inducing the development of the nervous system. So here's our neural plate. It's going to form an invagination and eventually form a neural tube. That neural tube is your central nervous system. The anterior end is going to form the brain, the posterior end is going to form your spinal cord. And you'll notice it's hollow for the ventricles and the canal. Cells beside it right here, and you can see them budding off the neural plate. Those cells are called neural crest cells. While the neural tube forms your central nervous system, the neural crest cells form your peripheral nervous system. All the neurons, sensory receptors, ganglia, all those neurons are formed from neural crest cells. They do also form the adrenal glands, for instance. What's left? Covered all of that. Oh, our last topic. We're gonna get through really good, which is okay with me. Too. We're covering all the material. You're asking all your questions. So, maternal changes during pregnancy. Now, this is drawn from different parts. Even some of this comes from chapter 28. So we're just going to look at some maternal changes that occur during pregnancy. First of all, at 2 months, the placenta is fully formed and begins functioning as a placenta. The placenta produces hormones. So throughout pregnancy, after the placenta forms, we're gonna have it produce estrogens, progesterone, and HCG. The progesterone and HCG, you know what it does, maintaining the uterine lining and whatnot, okay? The estrogens, which normally we saw go up and down, when you become pregnant, they go to heights you've never experienced, levels that a female has never experienced, because they go to very high levels because you have a large placenta producing a large amount of estrogen. Relaxin is produced by the placenta. It's a hormone that targets fibrocartilage, especially in the pubic symphysis, to allow it, normally it only moves slightly, it becomes much more movable. And that allows the birth canal through the pelvis to open up to enable the head to go through. Now, makes it bigger and more flexible. It doesn't come all the way back, which ends up making a female after birth to have slightly wider hips.
So relaxin is produced by the
Placenta.
placenta too?
Yeah. Yeah, all of these are produced by the placenta. These are all placental hormones. And it's not even a complete list. Human chorionic somatomammatrophin helps prepare the mammary glands, you can see mammo in the middle, for lactation, regulates metabolism of the mother and the fetus to keep them in control and in normal range. So that hormone's recently been discovered and is produced by the placenta. So let's look at the abdominal cavity of a female near term. So this is the uterus, covering up that much of the abdominal cavity. Their organs are on the posterior body wall. They're still there, they don't move. So where is the bladder in this female?
Underneath.
Right here, underneath all of this. Would you imagine that the bladder could fill to its normal full range? No, which makes for frequent urination. And you can see why. Let's see, where is the small intestine? Oh, we see parts of it up there. Wow. Large intestine, parts of it up there. Do you think having compression on the large and small intestine makes for easy flowing of material through the digestive tract? Constipation would be a continual thing, I would think, just trying to get stuff through with all the external pressure on the digestive tract. Where is the stomach? Stuck way up here. Do you think you could fill the stomach with normal amounts of food? I would think not, which means a female then is eating many small meals as well as taking many small times of urination. This is a big deal. Any other organs we need to think about? The liver, of course, isn't going to go anywhere. Let's talk about physiological changes. What changes happen to the cardiovascular system? What specifically, from you've learned from chapter 20, might happen?
Heart rate goes up.
Heart rate goes up. What else goes up? Blood pressure too, because you've got to get a lot of blood flowing to that placenta. Do you think blood volume will change?
Yeah, cardiac output.
Cardiac output's going to be up because your heart rate's up, as well as blood pressure and the volume of blood. Any changes to the respiratory system? Well, what's the big change seen in this diagram?
It's up.
The diaphragm is pushed up by all this. If you don't have a baby down there, when you breathe, the diaphragm dome shape pushes the liver down. Do you think your diaphragm can push the liver anywhere? No. So you can't get as large a volume. The tidal volume goes down, right? Which means if you're going to get more oxygen than you were before, you need to have a breathing rate go.
So you get more dioxide, carbon dioxide in your blood?
No, because they're going to increase their breathing rate. Yeah. They're just going to have more gas exchange. Increased breathing rate, reduced tidal volume. If you were to artificially somehow reduce your tidal volume, you would start breathing faster because carbon dioxide would build up in your blood. Okay? Any changes to the urinary system? The kidneys are going to do their normal function because they're sitting on the posterior body wall, and pressure or not from a uterus is not going to affect its functioning, but the bladder has a decreased ability to expand. And digestive system, we already talked about that. Many small meals, constipation. So those changes are pretty significant, aren't they? Yeah. And someone's trying to call me. Don't know who that is. Okay, are we through? Oh, we have a couple of questions. We are through chapter 29. Like I said, it's a small part. What are two important functions of the placenta?
So oxygen.
Nutrients and respiratory gas exchange. What else?
Oh, look.
Sage said hormones, right? Yeah, hormones, hormone production. Those are very important functions, okay? So, exchange nutrients, respiratory gases, and wastes, and produce the hormones that affect the metabolic systems. What effects would a decrease in progesterone have on the uterus during late pregnancy? Oh, I forgot to mention. Oh, okay. Did I mention it? The uterus puts progesterone, no, it puts oxytocin, no, forget that. It's oxytocin receptors, not progesterone receptors. Okay, if you had a decrease in progesterone on the uterus, what would that do? Yeah, the uterine lining would break down and would be surrounding it. And so, lead to uterine contractions and possibly labor. If you have a decrease in progesterone for any reason, it can cause premature labor. Remember, oxytocin receptors are put on the uterine muscles late in pregnancy so that oxytocin can stimulate and begin labor. Progesterone just maintains the endometrial lining. Progesterone does reduce uterine contractions. The progesterone produced late in pregnancy, remember progesterone is produced by the placenta, is there to, because progesterone's high, the progesterone receptors cause the smooth muscle to not contract. Those receptors are replaced, they're taken off, and replaced with oxytocin receptors late in delivery as it gets near time for birth. Okay? But before that time, they put progesterone receptors, and that receptor causes the smooth muscle to relax, to not contract. Remove that and put a receptor that's going to cause it to contract. That's what oxytocin does. So progesterone tells the smooth muscle of the uterus to not contract. Oxytocin tells the smooth muscle of the uterus to contract. So if you remove the progesterone, just don't have progesterone, there's no progesterone to quiet the uterine smooth muscle, it could start contracting and possibly produce labor. That's the relationship of progesterone to labor. So, it's the end of the class. I thank you for your attendance and hard work. I know that you have worked hard, you've learned a ton, right?
Yes.
Everybody agrees you've learned a lot? How many of you think your brain's about ready to explode? Yeah. It's not. Your brain is not yet full. Don't worry about that. So let's talk about exam 5. If I can get through this. Come on. There you go. Exam 5 will be Tuesday during class time, and it will cover chapters 17, 18, 28, and 29. You'll notice chapter 29 has very few questions. Okay? 23, 23, 23, 11 for 80 questions. They're objective questions. They cover everything that's referenced in the material. The questions on those chapters are already up on Canvas. After class, I will make available a file that tells you how many questions there are on each chapter. On the final, this file says, and you can read it, you can come up and look at mine, or you can read it on Canvas. There's 75 questions on the final. 65 of those questions are multiple choice application questions. Some of them similar to questions I have had up here. They're all multiple choice, no diagrams. 65 application questions. There will be 10 random objective questions at the end. You'll have the first 65 application, and then you'll have 10 questions from those chapters. I won't tell you because I just pick those and make them up as I go. From the chapters we covered. We covered 13 chapters. Chapters 17 through 29. I believe that works out to be 13 chapters.
So what are those objective questions?
They're just objective multiple choice questions. No, none of the questions on the first 5 exams will be on the final. None. 0. I've never had that. These are all brand new questions. So there's 65 application questions. You don't have to study anything differently. I recommend between now and Tuesday, you focus on these chapters and be able to answer the objective questions on exam 5. You'll finish your test at 12:20 on Tuesday. I will go upstairs and immediately grade the exam. If you stay here, I'll have them posted around 1. You may then come up and look at the test and see what you missed and what sections you should study. You will end up having 5 of these sheets with a list of things you missed and didn't know for each of the unit exams. Between Tuesday and Thursday, don't just read a chapter. If you, let's say you got 80%, you knew 80% of the chapter. If you just reread chapter 24, you're gonna spend 85% of your time reading stuff you already know. Huh?
Well, we forget, that was at the beginning.
You can review it. You'll be surprised. If you studied it like I know most of you did, it's gone into long-term memory. Most of it will be there. But this stuff was never, that you missed, that was never in long-term memory. I recommend you study sections you missed according to these questions on the objective tests. That's the best thing you can do. Because when you study that stuff, you're studying stuff you didn't get. I remind you again, please make sure you check all of your lab grades and make sure they're all accurate. Okay? You need to make sure that every lab grade,
Oh, the one that they're posted already.
yes, posted on Canvas. Because I go to Canvas and download the whole file, get rid of all the columns except the points and the name. And those points have to reflect what you earned. Now, a couple of you had a reason for missing a lab practical. You have to email me to remind me to calculate the replacement points. There's a method and the lecture instructor doesn't. I don't know if any of those people are here. Any questions? Yes?
I have a question on the study questions for chapter 28.
If you have a question on the study questions for chapter 28, ask me after class. I'm talking about any questions about Tuesday and Thursday.
You said, 65 applications for the final and 10 objectives, that's
Yep.
75 questions?
75 questions. Exam 5 will have exactly 80 on those chapters. This weekend, you study, I get to make up tests. And any question I put on exam 5 should not surprise you. You know what to expect. You've gone through at least 4 tests.
Will you provide us with the same thing as sample questions?
I will be putting up example applications for previous finals. There will be questions up. Sure.
Thank you.
Where is gonna be by the mothers?
Say it again?
Where you gonna post the mothers?
The modules. Yeah. The questions, there will be the other chapter questions over at the end, at the very bottom. Any other questions?